Hi Year 11, welcome to our video on enzymes. What we want to try and do is we want to try and answer this inquiry question: how do cells coordinate activities within their internal environment and the external environment? And specifically what we're looking at for these next few lessons are conducting practical investigations to model the action of enzymes in cells and investigating the effects of the environment on cell enzyme activity through the collection of primary or secondary data. So we're going to be looking at those environmental factors, so temperature and chemical nature, pH and substrate concentration. We're going to do experiments on all three of those. We're also going to be using data and graphing to model the effects of enzymes on the environment, and right at the end we'll make a model of enzyme action. So starting with metabolism, this is the total number of chemical reactions occurring within a living organism. And they can be divided into two categories: anabolic, building up, and catabolic, breaking down. Each step of a metabolic pathway in cells is catalyzed by enzymes. So cells need to maintain a constant temperature and chemical balance so that those enzymes can work efficiently and those reactions can occur efficiently. So looking first of all at our anabolic reactions, we're building up large organic molecules from simpler molecules. And in this example, we're seeing starch being made from small monosaccharide units like glucose. So here, and you can see that it requires the input of energy. Okay, so we call those endergonic reactions. They require energy. When we're looking at catabolic, breaking down reactions, okay, so breaking down organic compounds to simpler ones, for example, proteins being broken down into their amino acids, which then makes them easier to absorb into the bloodstream. And you can see in this example, it's going to release energy. So just briefly here, reminding ourselves that energy in the cell is stored in the molecule adenosine triphosphate. When we break that phosphate bond, it releases energy that the cell can use, and then we can store energy in the form of ATP by adding that phosphate bond onto it. So here we have catabolic reactions transferring energy from complex molecules to ATP. Okay, so we're storing energy here. We do that when we break down simple molecules, okay, into glucose, amino acids, glycerol, and fatty acids. And then our anabolic reactions, building them up, we transfer that stored energy in ATP to help us make those complex molecules. Just briefly on protein structure, we'll go over this in much more depth in Module 5 as part of protein synthesis, but so far you might have been thinking protein in terms of its primary structure, like beads on a string, amino acids joined together, but that is just an amino acid chain. We have secondary structure where you start to form bonds between those chains or sheets, for example, or helices, and then when they start to form out into more complex structures, we have tertiary structure until we finally have the quaternary structure. And in this one, we actually have these four colors, four individual subunits making up a functioning protein. You don't need to worry too much about this now, just be aware that proteins have a three-dimensional shape, and that's very important. So what are enzymes? These are large globular proteins. Their shape is essential to the way the enzyme functions. The molecule on which an enzyme acts is called the substrate. And the enzyme fits together with the substrate at a very specific place on the surface of the enzyme called the active site. That's really important. You can't change the shape of the active site because the enzyme won't be able to bind to the substrate properly. It won't be able to catalyze that reaction. It won't work properly. Oops. So here we have an example of a lysosome, okay, a ribbon-style model. That's what this shape is representing, and here's the active site. Again, here in a three-dimensional model, the active site of the enzyme where the substrate's going to bind to. Here we have this lock and key model. This is what we often start with in biology. So we have the substrate, the thing that's going to be reacted on, and we have the enzyme here. And notice that the active site here matches perfectly with the substrate. It's going to fit in like a lock and key. This is really important. You should copy this slide, have this somewhere, be able to explain how this model shows enzyme functioning. So here we start with our enzyme with the active site. And here we start with the substrate. In this case, it's sucrose, which is a glucose and a fructose bonded together. And you can see their shape matches the active site. So the substrate binds to the enzyme, forming this enzyme substrate complex. The binding of the substrate and the enzyme places stress on that glucose-fructose bond. The bond breaks, releasing our glucose and our fructose. So we've got our products. They're released, and the enzyme is unchanged, and it's free to go back again, bind to another substrate, and repeat all over. Now, this is the model that has actually replaced the lock and key, called the induced fit model. So what we see here is the substrate and the active site on the enzyme. They don't exactly match, but the enzyme changes shape to fit to it. It's induced to fit as the substrate binds to it. And here again, we have our enzyme substrate complex. Then that helps break this bond, okay, and we get our products released. So if we're looking at the glucose one again with the fructose, okay, and the products are leaving the active site of the enzyme. Notice that the enzyme remains unchanged, so it can come back here all over again and catalyze another reaction. So why change it? Well, the lock and key model, it was initially thought that the active site is rigid and the substrate is reciprocally shaped to fit into it, just like fitting your key into a lock. But when we tested this model, the results led to the proposal of what we now accept as the induced fit model, which is based on the realization that proteins are not rigid. That active site is not rigid. It's not like a hard piece of Lego. And evidence suggests that the binding of the substrate to the active site induces the enzyme to alter its shape slightly to fit more tightly around the substrate. So talking about cofactors. So some enzymes have non-protein groups that bind with the protein to help form the active site. So things like vitamins, B12, metal ions like zinc and copper, these are called coenzymes or cofactors. The enzyme can't function without the cofactor. So some poisons exert their toxic effect by disabling cofactors and therefore inhibiting enzyme function. So heavy metals like mercury and cadmium replace zinc cofactors in some enzymes and inhibit their functioning. So if you want to know a bit more about that, you can look at Surfing 75, Enzymes and Inhibitors, and Surfing 77, Enzyme Deficiencies. Here we have our image showing a cofactor and our inactive protein. So this is what is going to be our enzyme, but without that cofactor, it doesn't work. Okay, once the cofactor binds to it, it activates the protein, and we now have binding site available for our substrate to bind to. And just finally, a note on activation energy. So activation energy is the energy you need to put in to kickstart, get a reaction to happen. So reactions don't just happen, paper doesn't just burst into flames on your desk, and things don't just break down. You need a bit of energy to get it going. What enzymes do is they lower that amount of energy needed. Okay, so here we have energy on the y-axis and the reaction happening here. So we have our reactants. Without the enzyme, it needs this much energy, this much activation energy to get that reaction to happen. Okay, then the reaction occurs, you have your products forming, and the overall energy released is shown here. With the enzyme, now look at the difference. Here, this much lower activation energy required, and the reaction proceeds, you still get the same products, you still get the same overall energy released. So what is the role of enzymes? They can speed up a reaction without a change in temperature. This is important because heat damages living tissue. It lowers the activation energy needed to start a reaction. They are substrate specific. One enzyme, one substrate, that's it. Okay, and it's all to do with that active site shape being reciprocally shaped to the substrate, or you might see it referred to as substrate specificity, okay, or enzyme specificity. It has the matching shape for the substrate, so only that particular substrate can bind to it. The enzyme is not chemically changed or used up in a reaction, okay, so once the enzyme's done its job and the substrate's no longer bound to it, it can then go and catalyze the next reaction. Enzyme-controlled reactions are also reversible. So we're going to take a pause now and complete the Titan worksheet, the role of enzymes in metabolism, or your teacher might ask you to complete surfing worksheet 70 enzymes. But here are some key points I want you to think about just before you go and do those sheets. So remembering enzymes are a type of protein that speeds up biological reactions. The enzyme's not changed or used up. They're very specific. The area on the enzyme where it binds to the substrate is called the active site. It has a particular shape and it will only fit that substrate. And enzymes are affected by environmental factors, so changes in temperature and acidity, and those changes can denature enzymes. That's a word you might not have seen yet, denature enzymes. So what that means is the active site is changed so that they do not function. If they're changed too much, they might be irreparably damaged, and that's it, the enzyme won't work at all. So off you go, have a go at that worksheet, and we'll come back and look at the next section.